So I became interested in plants during my PhD, which was in neuroscience. I became interested in how cellular processes work in model systems that have different biophysical properties and use different evolutionary unique machinery. So that's kind of how I became a plant biologist. So I work on a process called endocytosis in mammalian systems, in yeast systems. It's incredibly well characterized. And in plants, it's been a little bit neglected. So basically, I saw the opportunity to kind of investigate it using a different model system and actually find some new stuff. And what's been really exciting is all of our predictions about how it was working have turned out to be somewhat incorrect. So it's quite an exciting model system to be working on. And it's also a real challenge because I do a lot of microscopy, and plants have always been considered quite a challenging optical sample, and we've basically been working to overcome these challenges and have imaged quite a lot of plants. I am now in a position where I have a bit more independence about what I'm focusing on, so I can kind of lead the research directions that I want to, and I think that's really been the most satisfying part. I mean, it's been great working with people in the past, but it's always nice to lead your own direction. I think you have to realize if you enjoy the basic discovery, because science can be a very challenging career, but it's also an incredibly rewarding one. It won't always go smoothly. You have to have a lot of resilience, and if you enjoy the basic science, then I think you'll be okay. So make sure that's what you want to do. The most rewarding part and interesting part has been, so a lot of the microscopy we've been doing, there's been some beliefs in the field that some of these things would be impossible, but coming from a naive mammalian background and working with the right experts of physics and optics, that we just kind of went ahead and did these things. And I think that's, yeah, don't worry if people say things won't work in plants, because sometimes they do. What we're trying to do is we're developing new types of ways of imaging the plant, and we just got some funding to do a lot of optical biomechanical imaging. So I think one of the things people have tended to overlook a little bit in the past with plants is the cell wall and its actual effect on the cell biology. And I'm hoping with our new technologies, so we're using spectroscopy to do correlative biomechanics and fluorescent microscopy, that we can start to look at the real biophysics mediating the cellular processes. I think the cell wall's role on plasma membrane organization and function has been a little bit neglected, and it's because it's quite a hard object to image at the same time as the membrane. So I'm hoping that we can really look at this dynamic interplay between the cell wall mechanics and the underlying physiological processes that it regulates. So we're interested in the biomechanics of the cell wall. And the way we image this is using a contact-free optical technique called Brillion light scattering, which basically we can use photons and how they're scattering off a material, determine how hard or how soft or elastic this material is. Because if it's hard, you get different scattering to photons coming off a soft object. I think some of the most exciting collaborations have come from physicists. My last postdoc was actually in the medical school of Vienna. So we were doing, working with medical physics to image people, and actually a lot of that is translatable back to plants. So I think, again, it's great to look outside the plant field to see what other people are doing, to see how we can adapt and adopt these kind of techniques into our field. We haven't learned so much that we can translate back to humans, but one of the key areas of my research is to understand the fundamental principles underlying eukaryotic processes involved in membrane trafficking, particularly endocytosis. So there's been a big debate in the field as to whether actin is involved in endocytosis based on work from yeast. And so it's always been assumed that in plants with high turgor pressure, you'd also have to have actin involved, but we found that that wasn't true. So we can actually then begin to understand more about how membrane bending and endocytosis processes work in mammalian systems by studying plants.